That's good. Okay. All right. It is the 11th of September, 2025. Is it okay if we write our homework answers digitally on our tablets, or do you want them on a piece of paper? I am completely fine with them being written on a tablet. I was trying to communicate that by saying penmanship, um, but I'm sorry I didn't. Yeah, but yes, if you want to write on a tablet, you're more than welcome to. I just don't want like typing something up and copying and pasting it. Absolutely. Okay, so um yeah, what we're going to go ahead and get started with is we have been talking about amino acids and protein structure. Now, amino acids are your building blocks, or in many cases, your alpha amino acids, your proteinogenic amino acids are the amino acids that are used for building up proteins. Now, I'm going to load this up. Um, and what we've primarily been focused on is the primary structure of a protein. Your primary structure of a protein is simply the amino acid sequence, starting from the N termini, reading amino acid, amino acid, amino acid, amino acid. What is the sequence? Alanine, glycine, leucine, isoleucine. What is the sequence of amino acids? Every protein is going to have a different amino acid sequence. Now, across organisms, you might have some similarities. You might have sequences that are very, very similar to one another. You might have some that are pretty dissimilar from one another. So that's all important information, especially when it comes to analyzing a protein for its function. Now, your primary sequence is that, or your primary structure, and I'll use those two terms interchangeably, sequence and primary structure. I'll use those two terms interchangeably. Your secondary structure is a little bit more organization. Now, the way that I always try to think about it is if you live on a street, your house might be next to your best friend's house, which is next to their, cousin's house. That's a sequence. That's like your primary structure of your protein. Now, if you were to zoom out from that, you might zoom out and see, oh, you live on a cul-de-sac. Okay. So your cul-de-sac would be similar to the secondary structure of a protein. If you zoom out even further, that cul-de-sac would be a local structure within a larger neighborhood. Okay, so that's I think that your neighborhood at that level would be kind of like your tertiary structure of a protein. So your secondary structure is often referred to as your local structures. The primary local structures, sorry, the most common local structures or secondary structures that you'll talk about are shown here. This one right here is an alpha helix, and this one right here is known as a beta sheet. Alpha helix, beta sheet. Those are the most common forms. Now, what's significant about them is that they are stabilized by backbone hydrogen bonding. We had a question the other day that was about, oh, well, what if an R group interacts with a backbone? That can happen, but that's not a part of the secondary structure. Your secondary structure is stabilized by your backbone hydrogen bonding. Okay. Now, these are some of the other things. Does anyone have any questions about this chart right here or this graph, the Ramachandran plot? Give me a one if you're good, or a two if you're curious. What sort of questions would you ask about that? Awesome. Okay, so we'll move on from there. Alpha helix. This is one of your major um, secondary structures or local structures of a protein. And this is all about, and lots of questions that I will ask about alpha helices relate to math and relate to kind of the rotations. Like I said the other day, one important way to look at this is to think about it as a spiral staircase. You walk up that spiral staircase. If you walk up, how many amino acids were required to get from the starting point to, the point immediately one floor above? Well, that would be 3.6 amino acids. Okay, so that's where one complete rotation requires 3.6 amino acids. The next one is, well, how high are you from the ground level to that next level? That would be 5.4 angstroms. 5.4, the character for angstrom looks like that. Now, last but not least is, well, what's the measurement of an individual amino acid? Well, that would be about 1.5 angstroms. And whenever I say in all directions, I mean, well, going up and going back and going to the right. So we're dealing with this in three dimensions. So an alpha helix or the average alpha helix is roughly 10 amino acids. Can an alpha helix be longer than that? Yes. Can an alpha helix be shorter than that? Yes. The important part about, or one important thing to think about with respect to an alpha helix is, well, what are the different parts of this molecule that are interacting? And I'm just going to pull my email client up. Okay, great. Well, can I do an example math problem? 100%. Okay, so I'm going to give you the setup to this problem, and I'm going to use the annotate tool so I can type it in. Disconnect. And then extend this a little bit. A plasma membrane is approximately 50 angstroms wide. How many amino acids would, actually, let me rephrase that. Okay, so everyone, can you visualize that 50 angstrom wide? Okay, so now I'm not holding my hands 50 angstroms away, but just imagine a width of this unit of measurement. And if you want, you could call it 50 feet, whatever. But that 50 angstroms is the unit that we use. 50 angstroms wide. What is the minimum number of amino acids within an alpha helix that are required to span that entire membrane width? Okay. Now let me try to move my question. Sorry, it's okay. Can everyone read that question all right? All right, great. Now, what this question is really asking you is it's applying something that's relevant. So imagine that we have a protein that is found, that one part of it is found on one side of a plasma membrane, and then the other side is found on another side of the plasma membrane. And we have an alpha helix that forms to get this protein through that membrane. So what we need to figure out is, well, how many amino acids are required for that? Now, I think the way that I would recommend approaching this is to say, all right, well, my width is, you could kind of set this up as a dimensional analysis problem. 50, sorry, my pen was being on the fritz. 50 angstroms. Okay, so that's the width. Now what I want to do is I want to relate, this question is asking about the minimum number of amino acids. Okay, so how many amino acids will span that? So what number do I need to use? I need to use a number that relates that width measurement, that 50 angstroms, to a single amino acid. So I'm going to use the one amino acid, 1AA, is, I saw this on the previous slide, 1.5 angstroms. Can you give me a 1 in the chat if you follow that selection? Okay, so that's my connection where I'm relating a measurement to a number of amino acids. So now I'm going to say 1.5 angstroms is 1 AA. So essentially, to solve this problem, I just have to do 50. Boop, 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 boop. Okay. Calculator. 50 divided by 1.5. That would be my answer is 33.333 and so on. So, your answer would be 33.3 amino acids. Can you give me a 2 in the chat if you follow that question? Okay. Now, this is definitely a question that I could alter. And another way that I would alter it would be something like, okay, here's the width of this membrane, and I would change the question to how many rotations does this helix make? Okay. Well, I would again go with 50 angstroms. And now what I'm going to do is I'm going to relate that measurement to the number of rotations. So 50 angstroms. Well, I know from my previous slide that the height of one complete rotation is 5.4 angstroms. So now I'd say 5.4 angstroms is one. I'm going to say ROT. I mean rotation, but 5.4. Okay, so now I would do 50 divided by 5.4. And that would be 9.25 rotations. Can you give me a 3 in the chat if you follow that calculation? Great. So these are the ways that I'm going to ask that sort of question. Do we need to memorize these values? Yes. You need to know the 5.4, 1.5, and 3.6. Are we allowed to have a little calculator with us to do the little math calculations for the test? Yes, you can. Where did I get the 5.48? I didn't get 5.48. That's my handwriting. That's 5.4 angstroms. So that came from right here. Um, Now, with that said, will our test be multiple choice or short answer? They will be multiple choice as well as some multiple select questions. Very similar to what you're seeing in the practice exam, which I added, I think, 5 more questions to the practice exam yesterday. So if you've taken it previously, go back. There's a total of 15 questions now. Okay. So these measurements are important for, this sort of scenario. A plasma membrane has this measurement. Okay. Why do we use 5.4 and not 3.6? That's a great question. We use 5.4 because what we're asking about is the width of that measurement. It's almost like how many joules would you have to stack on top of each other to span this region? And that region would be 5.4. Well, how many joules? If each joule is 5.4 angstroms tall, we would need 9.25. The 3.6 is not a measurement. That is instead the number of amino acids that make one complete rotation. Should we memorize the measurements? The 5.4, 1.5, and 3.6? Absolutely. And you guys can use a 3x5 index card or scratch paper on your exam, so make sure that you put those in. All right, that would be a good thing to write down on there. But this question at the top is the sort of question that I like to use. So if we were given the number of rotations, we could use that to find the number of amino acids. Absolutely. That's a great question, Melanie. So if I said there is an alpha helix with 22 rotations, how many amino acids does that consist of? Then you'd say, okay, 22 rotations, one rotation is 3.6 amino acids, 22 times 3.6, this. Alternatively, well, how large is this, or how tall is this alpha helix? It's got 22 rotations. Each rotation is 5.4 angstroms. 22 times 5.4 is this. Okay, I think I've addressed those questions. If anyone has any more questions, or because we are using Proctorio, will we just show it the index card before the exam? Well, how will that work? That's exactly what you'll do. The first question on the exam will prompt you to hold up each side of your index card, which you can use both sides of that index or note card. Hold up both sides for about a 3 count so that I can see it and so that you can prove that you had it. And then I'll go back and scrub through and I'll look at that. Okay, now I'm going to go on. I got to clear that because I don't need that marking on there. Done. Now, there we go. There we go. Okay, so this is the sort of question, and here's some more examples. Now, next up, a helical wheel. This is simply a way of displaying the orientation and organization of different amino acids in an alpha helix. The very first amino acid, sort of the N-terminal amino acid, would be amino acid number 1. And this is really trying to visualize and show you, well, where amino acids are whenever they are all kind of wound up into an alpha helix. So we've got amino acid number 1, the N-terminal amino acid 2, 3, 4, 5. Can you give me a 1 in the chat if that makes sense? Okay. Now, what this is trying to do is it's trying to take your primary structure, which I mentioned the other day that 1 knot indicates primary, your primary structure, and translate it to a secondary structure so that you can look at the primary structure of your sequence A, G, I, L, S, T, A, that primary structure of a protein and say, all right, well, what does that look like whenever I make it an alpha helix? Well, here's what I'm going to do. Give me a 1 if you understand and can follow that amino acid number 1 in this peptide is A. This is number 1. Okay. Now, what I'm going to do is I'm going to convert this peptide right here to a helical wheel organization. So amino acid 1, A. Amino acid 2, G. Amino acid 3, I. I'm going to scribble out these numbers as I'm doing it. Amino acid 4 is an L, 5 is S, and then could anyone tell me what is amino acid number 6? What letter should I put next to this 6 right here? T, a threonine. Okay, so what I'm wanting you to visualize is this is kind of the building up. So if you look at that 1, what I want you to think is imagine that you're standing at, you're at the top of a building and you're looking down at a spiral staircase. Amino acid number 1, this one right here, is at ground level. Amino acid number 15, that's at the highest level. That's closest to you. Can you give me a 1 in the chat if you follow that? Great. Okay, oh, great. Will they always be numbered, or should we assume the end of the red line is number 1? This color coding is really just kind of to help visualize it, just for like depth of perspective or perspective and depth of field. Um, but I would number them. And really, this is just like how would you translate this? Now, the significance of this is looking at the primary structure here. Amino acid, the second amino acid, which is a G, and this T. Well, in the primary structure, they're not right next to each other. However, when they fold up or they wrap up into this helical wheel, these are kind of on the same side. Now, that is significant because This helical wheel is showing the relative positioning of different R groups. And it's showing how the primary structure is one way in which different amino acids are close to one another, but the secondary structure, whenever this whole thing folds up, well, they're right next to each other, even though they might be far away from one another in the primary structure. The significance of this, what did you say, 1 or 15? 1 is going to be the farthest, 15 is the closest to you if you're looking down through the center of this. Great question. That was a private message, but a great question. So, the significance of this is you might have, like, let's say, you might have a clustering of amino acids that end up all being like these would potentially or hypothetically, these would all be polar uncharged residues. Oh, well, they're kind of peppered throughout the primary structure, but if these are polar uncharged, which I'm going to label it Pu, polar uncharged, and the rest are going to be non-polar, well, you actually might have two, or you might have a way in which different helices interact with one another. Because if you think about a polar amino acid, that's an amino acid that's capable of interacting through hydrogen bonding. So you might have two alpha helices, here's one and here's another, that they interact through hydrogen bonding. That's a significant part of protein-protein interactions. All right, now we'll proceed from here to introduce our next major secondary structure, and that is known as a beta sheet. Now, a beta sheet is just it's kind of your other major secondary structure. These beta sheets, well, they interact through backbone hydrogen bonding. The same thing, a carbonyl and an N-termini. N-termini and a carbonyl. Those are hydrogen bonds that are between those two different functional groups. Um, does that mean that the R groups cannot be next to each other? No, it doesn't. Well, do R groups, do they interact to stabilize a beta sheet? No, they don't. Now, what's significant about this is we've got two major types of beta sheets. There's a parallel and an anti-parallel beta sheet. On the left-hand side, you can see your anti-parallel beta sheet. On the right-hand side, you have your parallel beta sheet. Now, these two models, or these two drawings, are pretty accurate of what a parallel and anti-parallel beta sheet look like. Between these two, I'm going to label this A for anti-parallel and P for parallel. Can you tell me what do you think is stronger, the A anti-parallel or P parallel? Oh, this is great. I'm seeing a pretty even distribution of A and P. Now, I'm going to tell you, it's actually the anti-parallel beta sheet. The reason that it's the anti-parallel beta sheet is these two groups are directly across from one another. They are directly across from one another compared to this group right here and this group right here. In a parallel beta sheet, you have hydrogen bonds that are kind of at an angle. And with that angle, well, there's a risk of the length of them extending, in which case they would become weakened or even more weakened. Whereas whenever you have an anti-parallel beta sheet, you have these strong connections that are right across from one another. So anti-parallel beta sheets are stronger than parallel beta sheets. Now you might be curious, well, in these models right here and right here, which one of them is parallel and which one of them is anti-parallel? Are they both parallel? Are they both anti-parallel? The way that I've always kind of visualized it is I think about a peptide going in an N to C direction, N termini to C termini. Now, the important part about this is to say, all right, well, if we've got a polypeptide that looks like this, and this region right here that I'm going to shade, Sorry about that, everyone. This region right here, that is where our beta sheet has been established. Here's my N termini, and here's my C termini of my overall peptide. Now, this that I'm drawing an arrow to arguably is the C termini of this region of this peptide. Would you give me a 1 in the chat if you understand that? Okay, I got a question. Would that be some sort of steric strain to it? Yes, there would be steric strain to a parallel beta sheet. Okay, so I just drew this, and it seems like most folks follow that this is the C termini of this region of the peptide. That right here is the N termini of this region of the peptide. Here, what we have is, if you were to kind of cut this out and look at it, you would have a peptide where it's N and C, N and C. Would you agree with me? And if you do, add a 5 to the chat, that these two regions of this peptide are going in the same direction. Okay. Now, and I want to contrast that if you're like, I don't really see that. Well, let's say this peptide right here, this one right here. These two regions of a peptide are going in opposite directions. Give me a 7 in the chat if you follow that. Okay. So the question really is, are these going in the same direction? Are these regions of this peptide going in the same or in different directions? If they're both going N to C, then you've got a parallel beta sheet, and if not, then they're going in different directions, and they would be an anti-parallel. So the way that I always kind of visualize it is, and I always like to do very simple drawings of peptides. Sometimes my drawings are too simple and they're just kind of focused on what makes sense to Joel. And if I give you a drawing and I'm like, there you go, that's perfect, and it doesn't make sense, please don't hesitate to tell me. Okay, so we've got this right here, and I would highlight a region. This region would be a good indication of two peptide fractions going in the same direction compared to, that wasn't drawn well, compared to this right here and here, this region. So give me the first 3 digits of pi if you follow. And you can see parallel versus anti-parallel. Great, great. Okay, so what I want you to take away from this is anti-parallel is stronger because those peptide bonds don't have that steric strain. They are directly across from one another. Parallel, they introduce a little bit of steric strain. Their directions are the same, but it puts those hydrogen bonds at angles. All right, everyone. I feel like we're doing pretty well right now. And so we're going to go ahead and proceed. If anyone has any direct questions, please don't hesitate to message me. Can you try labeling the N and C on the beta images? Sure. Okay, so let me see. Just to clarify, parallel goes N to C, while anti-parallel goes to N to C and C to N. That would be correct. Okay, can I try labeling? Let me go ahead and clear these out. Okay, so you know what I'm going to do is this region right here. So this would be an N termini to C termini, N termini to C termini. N termini, C termini. Wait. No, Joel. Okay, N termini to C termini, N termini to C termini. Okay, so these images or these two regions that I picked of these would both be anti-parallel. Now, let's see. I don't have any combinations where it's parallel. Hmm, I've got to work on that. Absolutely. Thank you for your questions. Okay. So beta sheets. Now, there's two pieces of terminology. When people talk about secondary structure, they talk about primary structure, or sorry, they talk about alpha helices and beta sheets. Now, a beta sheet is a part, or sorry, a beta sheet is like the completed thing. A beta strand would be like this thing right here. So think about it almost like multiple beta strands assemble to make a beta sheet. I'm not going to ask you a question like dissecting those two terms and comparing and contrasting those two terms, but that's the way it's important to think about it. Now, a beta sheet and a series of beta sheets, they take on a couple of different structures. Parallel and anti-parallels have different strengths and differ in their stabilities. They can organize in complex and pretty elaborate fashions. For example, if you look at model A right here, now what I want you to do with me is kind of track them in terms of, I will, actually, I got to zoom out and then actually, oh no, I can do this. What I'm going to do is start at the end termini and go through this. And what I mean is just go imagining that I'm going amino acid by amino acid through the primary structure. It's going to go like this. Done. Okay, can you give me a Y or an N in the chat if you followed that? And you're zoomed in on this figure A, correct? Give me a 5 if you're zoomed in on figure A. Great. So these different regions of this polypeptide, they can be close and they can be far, and they can be close in proximity on the primary structure, but they can be very far from one another in the secondary structure. Now, what's significant about this is that these different structures, they actually are kind of found in different types of proteins. And oh, that's not what I want to do. Oh, that always happens. Anytime I try to zoom out, I zoom too far, and then I zoom to show all of my slides. But these pretty elaborate structures are used for different types of proteins. And can you find both A and B structures in a protein? Oh, that's a fantastic question. Um, Excuse me, I apologize. Yes, you absolutely can. So one of the cool things about protein structures is that you might have a region of a protein that has like a alpha helix followed by a beta strand or a beta sheet followed by an alpha helix. And then the rest of the protein might look like, and I'm going to introduce you to an awesome term, um, this region right here, the one that I circled right there, that is not an alpha helix or a beta sheet. That would be considered to be what's a random coil. So you might have a protein that's, let's say, one that's 700 amino acids long. The first 250 are alpha helix, beta sheet, followed by alpha helix. And it might be, you know, 7 helices that are 10 amino acids long each, followed by a beta sheet, followed by 7 more. And then the last, let's see, what would that be? 450 amino acids might be nothing but a random coil. Okay. Now, Melanie, I'm happy that you asked that question because that's showing like these proteins are complex. Now, in addition to that, as a protein gets more, oh, is the random coil made up of nonpolar amino acids? Melanie, fantastic question. A random coil, it can be It can be a mixture of non-polar amino acids and polar amino acids. Um, there's, I think the best way to answer that would be it's mostly going to be, I think, polar amino acids that make up random coilings. Some of those random coils might be because of, well, it's that's a very complicated question. And what I wanted to bring up regarding that is, um, can you give me a three in the chat if you've ever heard of AlphaFold, or a one in the chat if you've never heard of that? And if you haven't, that's totally fine. Okay, so for those of you who haven't heard about AlphaFold, AlphaFold is a program or a project that was spearheaded by Google. Now, this is something that people have been working on for 20 years, maybe even longer than that, trying to figure out what does a protein's three-dimensional structure look like based on its primary structure. So what does this thing look like? The reason is if you know what that thing looks like, then you have grounds to stand on where you can say, all right, well, I know that this protein has this particular function. I know that this protein with this function has this shape. You know what? If I were to design something to inhibit that protein's function, because I know what the protein looks like, if I were able to design something that fit right into that crevice and blocked it off, I think I'd be able to avoid some sort of, cancer progression, or I'd be able to avoid this sort of disease. So when you know the structure of a protein, then you know a little bit more of how it behaves. So AlphaFold was a project or a major project where they were like, all right, well, let's take the primary structure of a protein and guess what it looks like. And if we guess what it looks like, then we don't have to go through all of this work, or we can validate work, we can make earlier predictions. And when we do that, then we have a better chance of kind of fighting something off. Now, with that in mind, where I want to go with this is there are other secondary structures and they're a little bit less common. Your alpha helix and your beta sheets are your most common, but there are things known as reverse turns, also known as beta turns, and there are other helices that form that are not alpha helices. For instance, in the protein like collagen, in the protein like collagen. Collagen, for instance, is a protein that is a helical protein, but is not an alpha helix. But that's unique. That's an oddball because most of your other proteins are going to be alpha helices. Okay, now I was just talking about prediction. And back in the 1970s, there were two researchers, last names Chow and Fassman. And what they did was they put together a review article. And in this review article, this is in like 1972, I think. What they did was they said, "Hey, we've looked at all of these studies that people have done where they've determined what the 3D structure of a protein is. They know what this protein looks like." And what they did was they went amino acid by amino acid and they gave them scores. And they said, "Okay, well, um, here's what essentially what they would do is they would say protein number one. Well, it has three alpha helices and it's got two beta sheets. It's got a bunch of other random coils too, but it's got these three alpha helices and three beta sheets. So this is one protein that they would look at. And then they'd say, all right, well, let's look at the alpha helices. And when we do that, let's try to figure out the sequence. This is the published sequence of the proteins in that alpha helix. And they'd say, all right, well, protein one, alpha helix one. Does everyone follow me with my 1-1? Can you add a Y to the chat if you follow and an N if you don't? Okay. So they're looking at these proteins, and they're like, all right, protein number one's alpha helix number one. Um, Within that, we've got a we've got a glutamic acid, so I'm going to label that as an E, and then we've got a leucine, and then we've got an asparagine, an N, and then we've got a tryptophan, and the last amino acid in that would be, well, actually, we've got two more leucines. So they said, all right, well, in the first proteins, first alpha helix, that was the sequence. And then they went on to alpha helix number two, and they did the same thing. And it's almost like they got this very elaborate and very long Excel document. It's the 1970s, they didn't use Excel, but they went alpha helix by alpha helix, beta sheet by beta sheet, and they inventoried, they indexed every single amino acid that was in those different peptides. And they said, all right, well, what we see a lot of in an alpha helix is we see a lot of glutamic acids. So we're going to give that a score. We're going to give that a score. We're going to give every amino acid a score ranging from 0 to 2. And when we do that, this is what we see. And then, you know what? Amino acids we don't see very often in an alpha helix would be something like a glycine. So we give that a lower score. So long story short, based on data that had been already published, they came up with this algorithm basically, which gives you the ability to predict if a peptide forms an alpha helix or a beta sheet. And so they'd go alpha helix. Okay, what are the different amino acids within there? All right, well, there's a phenylalanine, there's a lysine, there's a leucine. Those are very common. And so what this enabled people to do was as soon as somebody discovered the primary structure of their protein, they could say, all right, well, the primary structure of my protein is glycine, glycine, glutamic acid, leucine, arginine, alanine, leucine, arginine, alanine. They would look at that primary sequence and then they'd say, okay, well, is this going to form an alpha helix? Can you give me a 9 in the chat if you follow what I'm saying? Okay, great. So when they're able to do this, they make this prediction and they say, oh, well, it looks like there's an alpha helix forming here. And, let's take it back a second. The examples with the alpha helix math, what did I have? I said something was 50 angstroms wide. Could anyone add to the chat what that thing was? A membrane. Okay. So let's say that you got the primary structure of a protein. You went through all of the different or all of the primary structure and you scored every single amino acid and you're like, oh, well, there's a bunch of amino acids that would gravitate towards forming an alpha helix. Interesting. Oh, you know what? Actually, there's about 105 amino acids 105 straight that would likely form an alpha helix. You know what? This could span a membrane. Huh. Maybe this is a protein that's associated with a membrane. Maybe this is a protein that's associated with importation of an ion or something like that, or of ions. Can you give me a 4 in the chat if that makes sense? So Chow and Fastman were instrumental in enabling people to take a primary structure and predict whether or not a secondary structure forms. This is extremely powerful because my computer just died, but does each score basically rosette? Yes, that's absolutely it. So the higher the score, the more likely something is going to be found in an alpha helix or a beta sheet. Now, there are some important details that I want you to know. Your alpha helix is going to prefer smaller and less bulky residues. Alanine, glutamic acid, methionine, leucine, phenylalanine. The less likely amino acids. So proline, histidine, arginine, glycine. These are less likely. Beta sheets, well, your most likely residues are tyrosine, tryptophan, phenylalanine, methionine, isoleucine, valine, threonine, cysteine. Your less likely residues are proline and glycine. Now, do I want you to commit these to memory? Nope. Do I want you to commit these numbers to memory? No, I don't. When it comes to specifics of secondary structure prediction, here's what I want you to take away. Okay, I got a private message question. How can both methionine and phenylalanine be more likely for both? Okay, so this is not a comparison. So that's a great question from a private message. The question is, or what they're seeking to answer. I'm sorry if I didn't articulate it clearly. Methionine has a 1.3 and a 1.14. The 1.3 indicates that it's likely to be found in an alpha helix. The 1.14 indicates that it's likely to be found in a beta strand or a beta sheet. Now, if you compare the two, it's more likely to be found in an alpha helix than a beta strand. But what we're really kind of looking at is, and this is a score of basically over the average. So methionine is more likely to be found in a beta strand than any other amino acid. And methionine is more likely than any other amino acid to be found in an alpha helix. Does that make sense? Okay. So when you look at these numbers, there are trends that I think are worthwhile to see. Leucine, for instance, it's got a pretty high value for both alpha helix and beta strand. Methionine, alpha helix and beta strand. When you compare the two of them, is it more likely to be found in one of those secondary structures? Yeah, it's more likely to be found in an alpha helix than a beta strand. But more than anything, what I want you to take away from these numbers is, hey, leucine and methionine, those are both amino acids that are going to, or there's a high probability that they will be found in a secondary structure. Lysine, by comparison, 1.23 and 0.69. Lysine is more likely to be found in an alpha helix, and it's not very likely to be found in a beta strand. Given the option, alpha helix or a beta strand, well, it's going to be found in an alpha helix. Given the option beta strand or random coil, probably leaning towards a random coil more than anything else. Okay, so what I want you to take away from this slide and these values is the closer they are to 2, the more likely they're going to be found in that structure. Then proline and glycine, these two amino acids, they're not very likely to be found in an alpha helix or a beta sheet. To pull back one more layer, well, these two amino acids are very different from one another. If you think about proline, I like to draw proline. I like to draw proline because when I was an undergrad, I could not draw proline very well at all. And it took me several years into graduate school before I could draw proline well. And not because, oh, I developed my art skills. No, it was just because I always screwed it up. The way that I think of proline is I think about it like a Monopoly house. NC. And then C, 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 done. So there is my, essentially, I'll add my carbonyl or carboxyl group down here. There is my R group for proline. And that's what it looks like basically at a a physiological pH. Now, with that said, this alpha carbon is covalently bound to that nitrogen, that N termini. And what that means is that basically this, where I'm drawing my X, is kind of anchored to that. So that makes this amino acid extremely rigid. It's almost like if I put my hand on my head like this, and I'm holding onto my hair, whenever I turn my head, I'm turning my arm too. My arm being the R group. Okay, so if I can't move my head, then I can't move my R group. I hope that makes sense. Give me a 3 if that makes sense, a 2 if I don't follow that, but I understand proline. Okay. So at the opposite end of the spectrum of proline, proline is super rigid, and I want you to remember that, is glycine. Glycine is very flexible. So it's actually too flexible. So when we compare proline and glycine, these are two amino acids that are both considered to be secondary structure breakers. These secondary structure breakers what that means is if you have, let's say, alanine, glutamic acid, proline, methionine, glycine, if we had this sequence right here, you've got this amino acid, this one, and this one, which they have a strong propensity for forming an alpha helix. But the presence of this proline and this glycine disrupt it. So they prevent that secondary structure from forming. So I want you to take away from this slide that proline and glycine are breakers. They are secondary structure breakers. All right. Now, secondary structure prediction is something that can be done. This is something that is, if you do a Google search for secondary structure prediction, then you can plug in a primary structure of a protein and it will give you a score. And it'll say this region is likely to have alpha helices, this region is likely to form beta sheets. It will give you that. Now, there's a lot that goes into that computation. There are certain residues that favor alpha helices, some that favor beta sheets, and others, proline and glycine, that disrupt it. Now, secondary structures are stabilized by backbone hydrogen bonding. The length of the peptide often matters because if it's not long enough and if it's less than 6 amino acids, you're unlikely to get that kind of hydrogen or backbone hydrogen bonding. Does anyone remember? Can you add to the chat? What was the length of the average alpha helix? How many amino acids did it take to make it the average alpha helix? 10. Now, 10 is not the minimum. That is the rough number for the average number of amino acids. So that means that there can be alpha helices that are shorter. That means there are alpha helices that are longer. Shorter than 6 amino acids, it's unlikely to make an alpha helix, which, think about that number, 6 residues. Approximately how many amino acids are in one complete rotation? That would be that 3.6. So if you only have 6 amino acids, at best, you're going to do like 1.5 rotations. Can you give me a 1 in the chat if you follow that and follow this length of peptide? Okay. Now, in addition to that, there are other factors at play. pH, whatever the solvent in the solution is, the ion strength within that solution can also influence secondary structure formation. There are tools that will help predict secondary structure, and that's very powerful because you can take the primary structure and predict, oh, does an alpha helix form here? Now, our next level of protein structure is taking those local structures and putting them into a larger molecule. That is known as our tertiary structure. The tertiary structure of a protein is formed by a single polypeptide. This is stabilized by covalent and intermolecular forces. For example, hydrogen bonding, hydrophobic interactions, and electrostatic interactions between different R groups. So one of the things that we've talked about is amino acid R groups. I've told or I've said since the, first or second day, you need to know your R groups. You need to know how they're classified because that's going to tell you whether or not an interaction is likely to take place. So, for example, disulfide bonds are also a contributor. So, two cysteine residues will form a covalent bond that those can be a part of a tertiary structure. Um, if I were to ask you what type of interaction takes place between, or actually, let me rephrase that. An ionic interaction is between which of the following amino acid combinations? And I'm going to number them rather than letter them just to avoid confusion. I'm going to go one, two, and three. An ionic interaction between D and L, then ASP and ARG, and then ALA and GLY. Whenever you're ready to answer that question, add your answer. We're talking about the R groups. Okay, so if we think about this, D and L. Now I mix this up so that it's asking you with the context of the three-letter and one-letter codes. D is aspartic acid. L is leucine. Aspartic acid is a polar negative. L, leucine, is a nonpolar residue. I'm going to jump to answer option number three. Three is alanine and glycine. Those are both nonpolar amino acids. Their R groups are going to interact through hydrophobic interactions. Then answer option number two. Aspartic acid, which is a polar negative residue, and ARG or arginine, which is a polar positive residue, those will interact through an ionic interaction. So number two is the correct answer there. Does that make sense? Can you give me okay. Yeah, Y works. And I'm happy to bring this up because this is definitely a sort of question that I will ask on exams. And I'll add some more of these on your practice exam. A little? Okay, so I guess. So let me ask another sort of question. H bonding. No worries. Sorry, I read it and I forgot forgot the question that I asked. H bonding will take place or will be between which R groups? Okay. So hydrogen bonding will be between which R groups? And I'm going to again give you a couple different answer options. Number one, I'm going to go with PRO and THR. Number two, THR and let's go with HIS. And then last but not least, I'll go THR and SER. Nope, not SER. That's not what I wanted. That's a good option. THR and Gly. Okay. So this is a good Joel Gray styled question. I've got one common element, threonine. Threonine is a answer option for all three of these. Now, with threonine, we know that that functional or that R group has an OH. An OH is a good group for hydrogen bonding. So then I need to look at the different or the other amino acid, basically. Proline. Well, I just demonstrated my great abilities that were acquired in graduate school that don't tell any of my undergraduate professors. Melanie, you're spot on. You're absolutely right. It's threonine and histidine. Proline, that's a nonpolar amino acid. Glycine, nonpolar amino acid. Histidine, given these three answer options, histidine's your best option. Now, if I had four, and I don't like to ask questions where it's like select the best answer. I want it to be a little bit more concrete than that, just because it saves everyone a headache, and this is a foundational class. This is not intended to be like, yeah, this is a foundational, it's an upper division, although a foundational class. If I had THR and SER, arguably that would be a better answer option than histidine, just because these both have hydroxyl groups. And that histidine, this one is a little bit more pH dependent because if that histidine is protonated, it's more than likely going to want to interact with an ion as opposed to something that is a hydrogen bond donor or acceptor. Okay, so that's one of the things that's important about R groups and knowing them. You have to know how they interact, how they change, and be able to say, like, okay, well, this group is protonated at this pH, therefore it will have a positive charge, making it a candidate to interact with a negative charge. Now, that's, sorry, that's what leads to our tertiary structure. So R group interactions. So now if we go back and we kind of inventory, primary structure is peptide bond stabilized. So what type of bonds stabilize these different levels? Primary structure is your peptide bond. Peptide bonds stabilize the primary structure. Secondary structure. I'll give everyone a bonus point on the final exam. No, on this first exam. If you tell me what types of interactions stabilize secondary structure in the chat. Hydrogen bonding. Now, this isn't like, this was not a, oh, but you didn't answer this. To make a more complete answer, or the best answer would be backbone hydrogen bonding. You guys got the point, though. Because you got the point. Um, then tertiary structure is R group interactions. So peptide bonds, backbone hydrogen bonds, R group interactions. Now, the important part about the tertiary structure is that it is all taking place within one polypeptide. If we go on above that to a higher level structure, well, we have what's known as the quaternary structure. Your quaternary structure is with two or more polypeptides. Two or more polypeptides, stabilized by covalent and intermolecular forces. So, so stabilization, favor, that's a great question. So stabilization can only occur between R groups. When it comes to the tertiary structure, so I'm going to jump back just a moment. When we talk about the stabilization of the tertiary structure, you have still the foundation of the peptide bonds. You still have the foundation of the primary and the secondary structure. So those forces still contribute to the stability of the protein. So the R groups will kind of drive like the global structure of that, though. Does that make sense? Okay. So none of these stabilizing forces are in a vacuum. It's like when you're talking about a tertiary structure or the 3D fold of a protein, that protein still has a primary structure, and it's still in part stabilized by that. Your quaternary structure, this is with two or more polypeptides. And that's where we come up with terms like monomer, dimer, trimer, tetramer, pentamer, hexamer, heptamer, octamer, nonamer, decamer. And I stop at 10 just because I can't remember the number beyond that. Um, but you have multiple subunits that fold together to give us, a big structure like this. This protein that I'm showing you here, what types of bonds can take place between R groups? Melanie, that's a great question. For the most part, our R groups are going to be limited to these. You have an ionic interaction, an ion dipole interaction, hydrogen bonding, dipole dipole, but there can also be hydrophobic interactions that drive tertiary structure. Quaternary structure, well, this is something that all of these proteins, these multiple proteins, can come together, they can carry out a function, and then they can disassemble. They can come together, disassemble, carry out a function. Okay, I think this is a good place to take a pause and stop for the day. I'm going to go ahead and stop my recording.